Welcome everyone to the session on FAIR in neuroimaging. Over the last 3 decades, neuroimaging research has seen large strides in the scale, diversity, and complexity of studies. There has been a significant increase in the open availability of data and methodological resources, alongside improvements in the quality of instrumentation. The awareness of rigor and reproducibility has increased with the advent of funding mandates and with the work done by national and international brain initiatives. This session will focus on the question of fairness in neuroimaging research. As a reminder, FAIR is a set of principles for making data and other resources findable, accessible, interoperable, and reusable. In this session, the speakers will be touching on each of the FAIR elements through ongoing efforts. With the amount of data coming online, search and filtering become critical. Through his talk titled Enhanced Dataset Search with Semantic Annotations and NIDM, David Keator from the University of California at Irvine will address components needed to solve this problem using the Neuroimaging Data Model, or NIDM. Valentina Borghesani and Julie Boyle from Université de Montréal will describe their experience of collecting and distributing a fantastic new resource that intersects artificial intelligence, human behavior, and neuroimaging. Their talk is titled The Village That Can Climb the Hill: Perks and Hurdles of Designing, Collecting, and Sharing Fair Datasets in Neuroscience. As many of you are probably aware, the brain imaging data structure, or BIDS, has made the lives of many researchers almost automatically fair, and created a community around it to keep extending the framework to support new domains of neuroscience. Melanie Ganz-Benjaminsen from Rigshospitalet and the University of Copenhagen will present open science efforts for PET imaging, from guidelines over BIDS PET to OpenNeuroPET. This is a coordinated effort within the positron emission tomography community to go from data acquisition to data distribution in a fair manner. Through her talk titled Open for Research: Challenges and Opportunities in Reusing Publicly Available Datasets, Elizabeth Dupré from McGill University will discuss how leveraging open data and computational platforms help address interoperability and reusability, but also means more effective science. And finally, the last speaker of this panel, Gustav Nilsonne from Karolinska Institutet and Stockholm University, will then take us on a discussion of ethics of FAIR in neuroimaging through meta-science questions around reproducibility and openness in science. After we have had a chance to hear from our speakers, we will take a short break and then continue with a panel discussion around FAIR in neuroimaging to talk about ongoing research and challenges faced by the community to enact these principles.
Hello, my name is David Keator, and I'm from the University of California, Irvine, Department of Psychiatry. I'd like to thank the organizers of the assembly and this session for inviting me to tell you a little bit about the work we've been doing on enhancing search across publicly available neuroimaging data sets using semantic annotations and the neuroimaging data model, or NIDM. This work has been supported by the NIMH, the NIBIB, and of course by INCF. So today I'll talk to you a little bit, give you a brief overview of the neuroimaging data model and what it is as a linked data format. I'll tell you a little bit about how we're formalizing data element definitions and descriptions in human neuroimaging experiments, how we're then using those enhanced, essentially data dictionaries to better annotate BIDS data sets with these semantic style annotations, allowing us to create NIDM records for enhanced query. And we'll look a little bit about concept-driven queries, how we're attaching concepts to selected data elements from these data sets, which really helps us perform queries across many data sets where the variables may be different with different properties. So the neuroimaging data model started many years ago. It was supported by the INCF Data Sharing Task Force. The idea was that we were going to build a linked data or semantic web format to capture metadata about your neuroimaging experiments. We built this format off of the semantic web technologies and the PROV family of specifications, where the PROV family of specifications is specifications for documenting provenance of things. And it's we found that we could use the same core structures to essentially capture information about experimental metadata. We found we could also use the same core set of structures to capture information about workflows applied to those data, and then the results that are generated. The idea being here that you could use a linked data technology to essentially store information about each of these steps in the life cycle of the data, and then use the query technologies that are built in semantic web tools to facilitate search across data sets. And so a basic NIDM experiment graph is pretty simple. It's got a structure where there's a project at the highest level of this graph. There are sessions that are part of the projects, and there are acquisition activities and entities, the entities being the object that holds the actual data or metadata about the acquisition. The activities are activities, they have start and end times, and capture essentially the provenance of that activity. So you can use these basic building blocks and you can create very complex graphs of your project. So the red box on the right is the NIDM experiment, a view of an experiment with multiple sessions, some imaging data that's associated with specific people or subjects, with some assessment data that's been collected. The blue box on the right shows that some of the derived data, in this case, it's a free surfer brain volume computation that was done using some of the data on the left. And so using these same basic structures of activities, entities, agents, and things. So once you build this RDF graph, you can serialize it to disk in a variety of text-based formats. These are not things that humans would really want to look at, but they're structured in a way that computers can use them quite easily. We like the turtle format, the red box on the top, because it's kind of human readable in some sense. Each of the little blue text that's a word and then a colon and then another word is actually a URL, and it expands out to a URL, so you can dereference it and find out precisely what was meant when the author put together this file. What did they mean when they were talking about a DCT license? Well, you could dereference that and go find out that precisely we're talking about the license of this thing and so forth. Another format that we like a lot is this Json Ld format. It looks like JSON, but it's the linked data variant of JSON. And so in the linked data variant, you have the keys just like you do in JSON, but there's this context file that's associated with it where we map the keys to URLs. So there we've described what we mean by each of the keys. Linked Json Ld also has the ability to store sort of more complicated graphs in the JSON structure. But beyond that, it looks like JSON, and it can be used similar to JSON. So what we found when we embarked on designing NIDM and trying to use it to annotate data sets, we found that oftentimes the data sets, even the data dictionaries for the data sets were missing, or they did not include some of the properties that we thought were important. A lot of definitions won't include units or mins and maxes, data type or value type, we call it. And so we, as part of our NIMH effort, we formalized the properties which we think are should be required and which should be optional when you go and you create a data dictionary for your variables. We call those variables personal data elements, because they're things that are used in your particular study, and oftentimes you decide what your units will be, what your frame of reference will be, what your categorical variables categories will be. versus common data elements, something like you might find in the NDA or NIH toolbox, where a group or communities come together and decided on a set of properties for those terms. And then we make a distinction with concepts, which are higher level terms. They wouldn't have a unit. They're not going to have a min and a max. They're these higher level concepts that we will use, and you'll see how we use them in the next series of slides. So once we decided on a set of properties, we went through and represented each term from the NIDM terminology, each term that we find in the BIDS standard specification document as Json Ld documents, which have the properties that we think are important in describing these data elements. Where we could not find them, we would ask the dataset providers or the BIDS community for a little bit more information, and this is still a work in progress. Then we went and annotated some data sets from Abide, ADHD 200, OpenNeuro, and started to do the association of selected data elements with concepts, these broader level concepts that we could then use to facilitate search. And so I'll tell you a little bit about how we did the OpenNeuro dataset annotations. The idea here being that we would pull the BIDS datasets from OpenNeuro. We would use the data dictionaries or the JSON sidecar files if they existed in the BIDS datasets, and we would add them if they did not exist based on interaction with the dataset provider, or in some cases things were obvious. And then we'd associate concepts with selected variables to facilitate search across data sets. So on the left is a DICOM structure that you might get off your scanner. On the right is the BIDS data set equivalent that I think all of you probably know about at this point. And a typical BIDS data set may have a participants.tsv file in the root directory, which is typically assessment data of some kind or summary measures from assessments that were collected as part of the study. The participant.json file is this data dictionary file that BIDS makes optional. Sometimes they're there, sometimes they're not. Our tools help you create these JSON sidecar files with proper annotations using the properties that we think are important. That they also let you annotate or attach concepts to those. And by doing that, you then get the ability to create this NIDM file, which has all this rich metadata that could be used in query. And so this is the a schematic of that process. We use Data Lab to pull the BIDS data sets. We use our NIDM tools to do annotations, where we search, COG Atlas, the Interlex, and some other places to allow you to select concepts to associate with your data set. Once you do that, your participant.json file is saved in your BIDS directory structure. It has many of the properties that we think are important. It also has this isAbout property, which are the concepts that help us link out and do improve search. Some of the command line tools are BIDS to NIDM and CSV to NIDM come with the PyNIDM libraries. You can take a look at those. Once we've done the concept annotations of the OpenNeuro data sets, we can look at concept use frequency and find that 66% of the OpenNeuro data sets had some measure of impulsivity based on our annotations. And then we can use these concepts to create a little search engine that operates just on the participant.json file. Also, we could do this with the NIDM files, which we are doing, but this particular demo is showing how to use the JSON sidecar files directly. Essentially select the concepts you're interested in, and you can then search across, at least in this case, the OpenNeuro data sets for data sets that have a measure of anxiety disorder and a measure of age. The ReproNIM effort has created what they call the Repro Lake, which is a graph-based database. These NIDM records for all these publicly available data sets that we've annotated go into this Repro Lake. The idea being this would be a place for a metadata search engine where you could go and search across lots of publicly available data find where there may be data that it's of interest to you in your particular scientific investigation, and then either download the data from us via the original sources, or if there are some requirements for, say, Irb approval or something, you can go off to the individual dataset providers and satisfy their constraints. There's also this GitHub repo where you can find our NIDM files of all these data sets we've annotated. You could also use the PyNIDM query routines locally to query across NIDM files. This works for small amounts of NIDM files, but it's not a full-fledged database or anything. But you can query for brain volume data if it exists in the NIDM files, the data elements that are in the NIDM files, information about the projects and subjects and so forth. You can't yet query using the concepts with this tool, but that's a work in progress. And so here are a bunch of NIDM, links to NIDM resources where you can find both the PyNIDM tools and information about our NIDM terms repository and other things. And I'd like to thank a lot of people who worked on these projects over the years. This is some of them. Thank you very much.
Hi, my name is Julie Boyle, and I'm the project manager for the CRIUGM project. And today, along with my colleague Valentina Borghesani, who's also a postdoc on the project, I'm going to talk to you about the village that can climb the hill, the perks and hurdles of designing, collecting, and sharing a fair data set in neuroscience. And we're going to particularly talk about how it relates to the CRIUGM data set. So during the talk, we're going to talk to you about the challenges of acquiring and sharing a fair data set. And I'm going to start the talk by talking to you guys about our village, that is who we are, as well as our hill, so what we're aiming for. And then Valentina is going to jump in and talk to you about the challenges of designing, collecting, and sharing a fair data set in neuroscience. So who are we? That is our village. The CRIUGM project currently consists of 42 researchers, so 15 professors, 6 staff, 5 postdocs, 5 master students, 10 PhD students, and 1 undergrad. And although the team is physically really located across 4 countries, because that's where the various labs that work on the project are, each one of the CRIUGM team brings in their own unique cultural diversity, and that's extra information that they bring into the project. In addition to cultural diversity, each one of the team members also brings in a unique academic background. We have team members that have trained in neuroscience, others that have trained in computer science, in psychology, in engineering, in mathematics, and physics. And here at NeuromOD, we definitely believe that both the cultural background and the academic background, as well as the general work experience, is one of the strengths of our team. So our hill. The CRIUGM project aims to train artificial neural networks to behave in a more human-like manner. We plan on doing that by acquiring and using extensive fMRI and MEG data that we're going to acquire from a small number of participants, using a variety of naturalistic tasks from a range of cognitive domains. Once we've acquired that data, we're really hoping to share that data set with the academic community, and Valentina is going to talk to you guys about that in a couple of minutes. The project itself is divided into 9 main branches. We have the platform, which I think a lot of the stuff we're going to talk about today falls under the platform. But we also have people working in vision, in memory, in emotion, in language, in video games, in audition, in modeling, and MRI. And I just want to point out that each one of those branches has their own sets of challenges and hills. So our hill, particularly as it pertains to the data acquisition, is exactly that, acquiring an extensive amount of data. Currently, we're scanning 6 participants, about 100 hours per year of fMRI and MEG data, and we plan on doing that over the course of 5 years. Now, what does that mean in terms of how large the hill is we plan on trying to climb? Here is a really lovely figure that was recently published by Nilsonne et al. This year, and where they've mapped various neuroscience data sets that are available currently in the community. And what we see is that on the y-axis, they plotted the number of subjects. On the x-axis, they've plotted the hours of fMRI data per subject. And what we can see is in the upper left-hand side, we have the UK Biobank, where they've acquired a little bit of fMRI data on lots of subjects. On the lower right-hand corner, we see the natural scenes dataset, where they've acquired about 40 hours of naturalistic image viewing on about 8 participants. And somewhere off the x-axis, you can find the OpenNeuroPET project. So that definitely gives you an idea of how large the hill is we plan on climbing. One of the challenges or the hurdles of acquiring such a large data set over a long period of time is to continue to assure that we can acquire quality data throughout the duration of the project. And one of the ways that we do that is that we're constantly challenging our standards of what good quality data means. And to come a little bit full circle, we do that with the help of our village. And definitely it's one of those instances where the village's both cultural diversity, personal work experience, and academic backgrounds really come in useful in order to be able to check ourselves to make sure we're staying on track. And of course, we also do that with the help of the larger community, and particularly community members like the ones here at INCF that help set standards and goals for what good quality data is. So thank you very much.
Thanks, Julie, for setting us on the right path. I'm Valentina, one of the postdocs with the team, and I'm here to tell you about our climbing adventure. And let's start with 3 things that need to be ready even before you put your shoes on. So first, get your Irb approval. I'm sure this comes with no surprise and that we all see the value and importance of a carefully drafted consent form. But the word of caution here is that it is always more complicated than you think, especially when sharing data is involved. In our case, as part of a public health institution in Quebec, we have to face strong restriction for data sharing. Our ethics committee follows provincial law, which is more restrictive than the federal one. So the suggestion is to consult with experts early on in the process and to check out some of the tools that the neuroimaging community has been developing precisely to help in these cases, such as the Open Brain Consent Initiative. So the second challenge is twofold: preparing experimental protocols you can share with stimuli you can share. Generally speaking, the options are 2. Either you find something that is already available or you build your own. And again, it's the community that will be your lifeboat. Reaching out and building collaboration will be the best way to have access to all the great data sets that are already available. And the silver lining is that whatever you will end up building yourself will be worth sharing, perhaps even publishing, and then other researchers will be able to benefit from it. So now you're all set up for your climbing adventure. You just need to recruit participants and acquire the data. A piece of cake. Your subjects need not only to agree with the current research plan, which in our case was already pretty challenging, but also with the idea that the data will be shared. So we were lucky enough to find 3 women and 3 men willing to embark in this journey with us and to be scanned for a total of 500 hours over 5 years. That's quite the commitment. And data acquisition itself is no joke, but quality data acquisition of a multimodal dataset you will want to release publicly is quite a challenge. There were many technical and practical issues to be solved, from the need to build an MRI, MEG compatible controller, to the fact that most of the cool video games rely on closed source code. And the whole process was pretty much a trial and error, requiring many iterations, and it's safe to say that the quality of our data is just getting better and better. The silver lining is, again, that you will be contributing to the community at large. Your seemingly side projects, your effort in working open source solution for your hardware and software challenges will be someone else's starting point. And one suggestion is to take detailed notes all through the process. a subject falling asleep, or even just not feeling great, a glitch in your stimuli presentation setup. Whatever happens, no matter how small, if it might impact the data, you should write it down. The future users were not in the scanner with you. Okay, so now you have acquired your data. So are you done climbing yet? Not even close. So first of all, we said we want to share quality data. So this means that one has to carefully check all data types, flagging anything that is of note. And ideally, compare the results to previously published work. And there might be cases in which you have to settle for something similar, but there's always a good enough metric or standard you can refer to. And of course, both the raw data and the pre-processed one needs to be well organized, not just to help the other researcher that will have access to it, but also your future self. The step of data curation is critical, and thankfully there are community-driven tools and standards that we can rely on. Speaking as a naive user, there might be a steep learning curve for some of these, but I promise they are worth every drop of sweat. Finally, you're ready to share your data. You will have to decide who has access to it and how. In our case, we opt for the register access option. So PIs with university credential can fill in a form on our website explaining what type of research will be conducted to ensure that it fits our general IRB. And then a data transfer agreement is signed and the scientific fun can finally begin. Please note that you can already declare your interest and be contacted as soon as we will release the first batch of data, hopefully late spring. So now you have reached the top. You can enjoy the view while thanking all the villagers that brought you there. And to find out more about our research, please check out our various online platforms. We got something for all your needs. In particular, I suggest you check out our YouTube channel, where we just uploaded one video per main ongoing project within CNeuromod. And before opening the floor to your comments and questions, please let me thank all the people behind CNeuromod, in particular the subjects and the scanning team that made it all possible, and Pierre Bellec and Basile Pinsard, who are really the core member of our platform. Our deepest gratitude goes also to the funding sources and supporting institutions, first and foremost the Fondation Courtois for sponsoring our ambitious project. And of course, thank you very much for your attention.
Thank you so much for having me present at this INCF session on fair approaches in neuroimaging. In my talk, I will tell you a little bit about the open science efforts for PET imaging. And we'll start out with the guidelines that were made for PET imaging over the new PET-BITS extension, all the way to a data sharing platform called OpenNeuroPET. So positron emission tomography has spiked some controversies in the last years. For example, in 2014, Paul Cummins published this commentary in NeuroImage where he basically asked, does the white elephant now soon pack his trunk? And his argument was that in general publication rates for PET were going down and fMRI was anyways taking its place. Well, this actually got him quite some answers from the neuroimaging PET community, especially Roger Gunn and Eugenie Rabiner made a very adequate response by actually highlighting that some of the search metrics that were used in the previous commentary to find and indicate PET publications was faulty, and therefore there were actually a lot more PET studies going on in the world than it seemed when Paul Cummins was writing his commentary. And furthermore, we also had a commentary following that which highlighted that with the advance of the new PET-MR machines, we actually should see more interesting and even more widespread use of PET again than we had in the recent years. PET is much more than clinical scans with FDG. So a lot of people look at PET as a clinical tool. It's a standard tool when you're going in for cancer screening to get a PET scan. Often it's a static scan with FDG. And while this is a very useful clinical tool, this is by far not the only tool that we can use when we're using PET. PET can visualize and quantitatively measure the function of biological and cellular processes in vivo. And so in this case, it can actually give us an insight into the metabolism of the brain or the whole body, but especially in the brain when we're doing neuroimaging on a whole different level than fMRI can. And already back in 2007, a consensus nomenclature for in vivo imaging of reversibly binding radioligands was published. Here, notably, Robert Innes was the driver behind this work. Then it has taken some time. After this consensus nomenclature, there was a consensus article for the radiochemistry. And additionally, at the neuroreceptor meetings in 2016 and 2018, a community effort was started in order to build a consensus on how to publish PET experiments. And the reason for this is that PET experiments are quite a bit more complicated, I would pose, than MRI experiments, because you have so many external measurements of radioactivity, maybe of pharmacological agents involved, that it needs to be very, very carefully described how the experiment was done. Otherwise, the experiment cannot be repeated. Bob Innes went back together with Gitte Mosknusen, the head of the neurobiology research unit, and myself, and we gathered a big group of about 40 people that are working on PET imaging in all the large centers around the world. And we tried to put together guidelines for the content and format of PET brain data in publications. And following this guidelines paper, the floor was basically made ready to also build a PET extension of the brain imaging data structure. And what is the brain imaging data structure? In short, for the ones of you that haven't used it before, basically it's a data structure. It has nothing to do with a, so to say, file format per se. It's basically about how to organize your data. It follows certain folder structures, how to name your files so they're very automatic readable, how to document your metadata, what kind of metadata should be involved when you report your studies. And then it uses community standards, meaning it is built on exactly things like the guidelines paper in PET, where the community has come together and said, okay, in order to reproduce PET images, PET imaging studies, we need to follow these standards. And you can see more of this there at the bitsneuroimaging.io web link. Now, specifically the PET bit standard has been very challenging to build. And the reason for this is, like I already mentioned, that in terms of the imaging modalities that had been included in bits before, PET is quite different in terms of the experimental setup. For example, in PET, everything is about time. We're dealing with radiochemistry, and here we always need to adjust for the radiochemical decay that we're dealing with. So we decided that within PET bits, everything should be in relation to a time zero, which could either be injection of tracer or the scan start. For some tracers, you would want to eject the tracer maybe 45 minutes, maybe an hour before you put the person in the scanner. This depends on what the permeability of the tracer is to the blood-brain barrier, for example, or how long distribution in the body takes for the tracer. Or it could also be scan start. Then one key component is, of course, the PET imaging file. The PET imaging file consists of different frames. Usually, like I already mentioned in the clinic, FDG scan would consist of several 5-minute frames. They would then be basically averaged, so you get static images. When you're doing more advanced kinetic modeling experiments, you're actually, or dynamic PET experiments, where you're employing kinetic modeling, then you actually have frames that vary very much in duration. They could start with only 5 or 10 seconds and then go all the way for about 5 minutes in the end, and could span a time frame of up to 120 minutes, for example. Especially if we want to validate new tracers, or in case we want to, for example, do a more advanced pharmacological intervention, then we also want to take blood measurements. And in these cases, we also have blood measurements by, for example, the autosampler or manual blood sampling. And this needs to be all synced up time-wise with time zero, which can be injection start or scan start. So how does this look like when we actually have it on disk? So the PET bit standard follows the regular bit standard. It has the meta information we all have, the bits version, then what is the name of the data set, where was it made, who are the authors, funding, what's the DOI of the data set. Then it gets followed by the PET relevant information, which is saved in a PET.json file. A JSON file is a simple structured text file. Here we know the manufacturer of the scanner, what type of scanner, in our case a Siemens HRT scanner, what body part was scanned, what are the units of our measurements, which can be quite important because not all centers use SI units. Then the tracer name, what radioactive tracer are you using, and then especially also the injected radioactivity. So all of this is in our PET JSON meta file. And then of course we have the actual image file. They are converted from the scanner formats, which is in Siemens, for example, the ECAD format. For other manufacturers, it's the DICOM format, to a NIfTY file. Then, if available, there will be some recording files. The recording files can be with manual blood samples or with autosampler blood samples. They will always come in pairs of a JSON file, again a structured text file, and a tabular.tsv file, again a tabular text file. Here we get some information on what kind of blood measurements are in the first place available. And then we will also know if some kind of correction was applied, for example, and we get detailed information about what the columns of our tabular file are. And then we have the simple tabular file, which is easily readable by the common modeling tools. For example, PMod in PET is widely used, or PVE-Lab that was developed at NRU. And those tabular files then give you all the necessary data that you need in case you want to do full arterial blood modeling. In most of the cases nowadays, we have an R image available together with the PET. In this case here, it's following the regular bit structure, so it lies under ANNET. It's a T1 weighted image, and it gives you all the details about this. Additionally, in a lot of our studies in our center, we're also requiring, for example, fMRI data. This would then be added to the structure in a func folder. What's the status of it? Well, we're quite far. We are having the pull request already to the main bit specification, and the last finalizations are done there. So by the time I talk to you, it might even be already merged. And an article detailing what PETS is about will also go out for review quite soon. So OpenNeuro is the official repository for the BRAIN initiative, and it's part of the Amazon Public Datasets project. It already has a lot of public datasets and thousands of subjects online, and there get new datasets uploaded every month. What we have worked on is now extending OpenNeuro to OpenNeuroPET. OpenNeuroPET will serve on the one hand as the place where you're using the bits PET format and you're putting your PET status, but also on the other hand, it will showcase best practices for how to process PET data and also help you to do quality control checks. In general, we're hoping that a framework like this will be able to educate the community, but at the same time, of course, we want always feedback from the community. What is useful for the community in order to really get everything they need out of the data sharing initiative? And a main goal of the project is also to establish average images of receptor density. At our side, we have already published an atlas for the serotonin system and also an atlas for the GABAergic system. And in this sense, we would like to, based on the data of healthy controls that get uploaded, provide the community with atlases of different receptors. The OpenNeuroPET setup, it consists of two major sites. Robert Innes at NIMH, who got funding through the BRAIN initiative. He is also closely collaborating with us and with the MGH, the Martino Center at Harvard, and Russ Poldrack is the consultant on the project, as well as the European side of the project, which is headed by Gitte Moos Knudsen, head of the Neurobiology Research Unit, And it's funded here by the Novo Nordisk Foundation Research Infrastructure Program. And here we're also collaborating with the American side and MGH, and have Russ Poldrak from Stanford on as a consultant. Thank you very much for your attention. Thanks to all my collaborators and colleagues. And yeah, I'm looking forward to the panel discussion.
Hi, my name is Elizabeth Dupré. I'm a PhD candidate at McGill University, and today I'm here to talk to you about challenges and opportunities in reusing publicly available datasets as part of the FAIR Approaches for Neuroimaging Research session. In general, I know there's a lot of ground that we could cover here, so I'm just going to touch on 3 different vignettes from my own work that highlight 2 opportunities with reusing open data, specifically around tool development and scientific research, as well as 1 challenge, which is having sufficiently available metadata for appropriate reuse. And before we get started, I know you'll hear this from several other speakers in the session, but I do want to acknowledge some of the caveats that we found around open data. Of course, many researchers do not yet feel comfortable with the technical concerns around data sharing best practices, and this is something that the community is actively addressing through education and tool development. But not all data can be made openly available, even with the best practice tools due to ethical or privacy constraints. Even so, we can still consider the impacts of open data on scientific research in communities with the data that can be openly shared. I also just want to highlight briefly that there are many different things we could be talking about when we say data sharing. So this is a really lovely figure from a paper by Poldrak, Gorgolewski, and Barroco a few years ago, in which they highlight 2 different dimensions along which we can consider data sharing, both potential for reuse and cost of sharing. And today I'm really going to be focusing on this bottom dimension of raw data, because this is what I've used most in my own work. But of course it has the highest cost of sharing, so it exacerbates several of the problems I've just touched on. Now, given that, let's go ahead and think about first how open data can drive ongoing tool development, scientific tool development. So one scientific software with which I'm quite familiar is the Python software library NiLearn. And NiLearn is a library for statistical learning with neuroimaging data. And in NiLearn, we really heavily rely on open data to generate our example gallery. So this is an online resource where researchers can see different examples of NiLearn usage in many real-world research contexts. So asking questions about classification of different tasks or decoding or general linear models, similar kinds of analyses, which researchers often want to do with their own data sets. So here, because this example gallery is really designed around being able to highlight different kinds of analyses that are possible with NiLearn, these different data sets that we include to support the example gallery are chosen for their didactic potential, and they're useful both to ensure first that the software is able to meet real-world use cases, so things that researchers really would want to do with their own data using the NiLearn library, and to also introduce community members to data analysis. So in many cases, researchers may come and they may not be familiar with a particular kind of analysis, but by looking at the example gallery, by playing around with the analysis with the openly available data, they can get a better understanding of how it could be in turn applied to their own data set and allow them to ask their own scientific questions. So I just want to show one example from the example gallery. So this is taken from an example where we look at classification of participants into children or adults in a developmental data set based on their functional connectivity matrices. And this is a data set I'm quite familiar with because I was able to add this to NiLearn for this example in part. And this data set is actually adapted, from one shared by Richardson and colleagues on OpenNeuro. So we were able to access the data set on OpenNeuro, process it with fMRI prep, and share the derivatives on open science frameworks such that they could be included in the example gallery. And this has been really, really instrumental in teaching these ideas. I know I myself have taught this particular example at a number of workshops now, and having this data available makes it so much easier to be able to really relate these concepts of both classification and deriving functional connectivity matrices, which are two things that I know researchers are quite excited about doing in their own work. So this data set has been really a force multiplier for these efforts. Now, I told you that we don't just need to think about open data sets in the context of scientific tool development. We can also think about them in the context of scientific research and enabling new kinds of scientific research. So here I just want to give an example from one research study that I was recently involved in. So recently, in particular with Thomas Basile, we wanted to compare the performance of several functional alignment methods. So for the purposes of this talk, it's not particularly important what these algorithms were or what our comparison metrics were, but what's important is that we did have in-house data available for testing. However, we were not interested in just using our in-house data because we know that benchmarking across multiple data sets provides more reasonable estimates of performance. So for example, if we look at the machine learning literature, we know that even small variations in data characteristics, so things as small as the particular scanner that was used, the particular scanner model, we've seen in neuroimaging research as well, can really cause significant changes in algorithm performance. And so it's really important to be able to look across a range of different data sets, different data sites, in order to get a more reasonable estimate of how a given algorithm will perform on average. And so we did a bit of combing through different data sets to find ones that were appropriate for this particular study. And I'll touch on a little bit after some of the challenges we faced, but I just want to highlight the 4 data sets that we were able to include, all of which are large initiatives, which have been used with it in themselves many times over, but having them available here to combine really enabled us to ask more deeply about these algorithms in a way that would not have been possible with a single data set alone. So here I just want to show really quickly an example figure from a recent preprint we put out. But what's really important is that each dot here is a different data set, a different task from a given data set. And what I really just want you to notice is that there's quite a distribution. So if we had only taken a single data set, we might have missed some characteristics of this distribution that are possible to see when we look at all these data sets together. And this is something that just wouldn't have been possible if we had been restricted to data that we had in-house, because having this range of data characteristics is really quite difficult to achieve without open access resources. So hopefully I've convinced you that there are a lot of really exciting opportunities available with reusing openly available data. And I do want to touch on one of the major challenges that we've encountered, which is having sufficiently available metadata. So in general, finding appropriate, well-described data sets remains a challenge. And this is because every scientific question requires something slightly different. And in general, it's hard to know as a researcher what kind of description you need to give of your data to help someone else answer a question that you don't know that they're going to ask, right? And so we just want to give as general of a description as possible. And this is where standards like the brain imaging data structure or the neuroimaging data model are really, really critical to be able to help us provide these standardized descriptions. Of course, there are many other INCF data standards, but these are 2 that I'm quite familiar with from neuroimaging work. One challenge that we've in particular faced is when we're thinking about naturalistic neuroscience, that is neuroimaging data sets that include naturalistic stimuli such as recordings or movies. Finding data sets with fully available stimulus information can be really quite difficult due to copyright restrictions. So I told you in the beginning that some data cannot be shared due to ethical or privacy constraints, and I know you'll hear more about that later. But similarly for naturalistic stimuli, copyright restrictions can make it really difficult, if not impossible, to share data stimuli in certain jurisdictions. So we, in a recent perspective piece, had a bit of time to think about some recommendations in general for sharing naturalistic stimuli, and I think they relate quite nicely to considerations around open data in general. So first, if you can, much as with open data, directly sharing stimuli allows for the broadest potential for reuse. And as we've seen with open data, the reuse possibilities are really quite exciting. And so if it's possible to share the stimuli, much as the data, then that's really helpful for the field. In some cases, however, that may not be possible. And so using public domain stimuli or reusing stimuli that have already been publicly shared can be really useful in order to make sure there's availability of the stimuli. And in some ways, this also acts as the point of benchmarking data sets for a field. So being able to analyze the same data set or the same stimuli across multiple groups enables us to get a better assessment of both the characteristics of that particular data, as well as how different algorithms or different learning methods interact with it. And finally, much as with data itself, the importance of metadata is so crucial, even more so in those cases where you can't share all details of your experimental setup, the stimuli itself, or the data. And so the metadata for naturalistic stimuli is not yet incorporated into data standards like BIDS or NIDM. And so for now, we recommend using reporting guidelines such as those from van der Waal and colleagues. But of course, this is another direction in which standard development is always continuing and will continue to aid in reuse. So with that, I just want to thank my supervisor, Dr. Jean-Baptiste Pauline, as well as my lab, our collaborators, 2 datasets which have been really instrumental throughout all of this work, my funders, and you for your attention. And then I just want to leave with some take-home ideas. So thanks so much.
Hello, I'm Gustav Nilsonne. Thank you for having me in this session. I'm going to talk about the ethics of FAIR in neuroimaging. The outline of my talk is going to be as follows. First, I will briefly discuss the concepts of FAIR versus open data. I will then talk about weighing benefits against risks and harms for our participants. I'm going to talk about assessing re-identification risk and the sensitivity of data. I will briefly discuss synthetic data as a possible solution. I will talk about recommendations for information to participants and end with a few discussion points. So, why are we talking about FAIR data? If we go back, let's say 10 years, I think many people were discussing a vision for the future of scientific data involving large open datasets available freely to download for anyone. In the ensuing debate about protecting participants' integrity and privacy, the emphasis has shifted, and the concept of FAIR, as you know, is often described with the phrase as open as possible, as closed as necessary. So what we envision in a FAIR ecosystem as opposed to an open science ecosystem is rather a set of connected services that have appropriate safeguards for the protection of personal information, but which also have features such as clear licensing, machine readability, and access mechanisms that allow researchers to find, access, and reuse research data, including data with personal information, under such restrictions as may be necessary. And we do need data to be accessible for reuse. I'm not going to spend a lot of time on this point because I think it's not necessary to convince anyone in this session, but I will just briefly touch on 3 categories of reasons why data sharing is valuable. First of all, that it enables cumulative science. It makes it possible for us to put together different data sets and achieve higher statistical power to build on earlier work rather than having to collect new data to answer a question. And thereby it can increase precision and reduce bias. A second reason is that we can make new discoveries in data that already exist. For example, one of the things that I've contributed to that I'm most proud of in the last year is a reanalysis of a dataset that I had posted on a repository where the group of Yuri Hasson in Italy were able to reanalyze associations between eye tracking measures and resting state functional connectivity and draw some very interesting new conclusions without having to gather a new dataset. And lastly, critical appraisal. If the data are open, then we can interrogate claims that are being made in the literature. We can, for example, reanalyze data and check assumptions that were made in the published analyses, and so on. A general principle of research ethics is that we need to weigh benefits against risks and harms. And that's one of the reasons why I wanted to briefly touch on a few benefits of data sharing, even though it may be obvious. So, when we have participants in our experiments and they undertake certain risks and harms, the harms of neuroimaging are typically quite limited, but nevertheless they exist. And the risks to participants' privacy are there if they can be re-identified in the dataset. Now, these risks and harms need to be outweighed by the benefits in terms of knowledge gain. And I want to stress this point because it is not uncommon to find, for instance, data protection officers who assess risks and harms and stop at that point. Without assessing the benefits. So we have a duty to our participants to protect them and their privacy, but we also have a duty to our participants to make sure that the greatest knowledge gain can be obtained as a result of this participation in our research. If we protect the participants' privacy fully by throwing the data out the window, then we are doing our participants a disservice because the knowledge gain can now no longer outweigh the risks and harms to which they have agreed in their participation. In the following, I'm going to talk mainly about risks that are due to re-identification of data, because this is a very salient point in discussions of data sharing. I'm not going to talk about other risks, such as the risk of incidental findings on an MRI scan. How can we think about assessing the risk of re-identification? Well, one framework that I like starts off with the notion that the risk depends on the likelihood of re-identification and the sensitivity of the data. This means that if data are less sensitive, then we may be able to accept a somewhat higher likelihood of re-identification. So we need to consider what it is that is sensitive in the dataset. What kinds of data are included? Typically, health information is a large category of sensitive data. Under the General Data Protection Regulation, GDPR, health information is always sensitive and includes nearly everything we can measure from a human. There may, of course, be other sensitive data in our datasets as well, like sexual orientation and genetic information. Sometimes we can reduce the risk by removing sensitive data. For example, in one study I did a long time ago, we asked participants about their sexual orientation, even though it was very tangentially related to any of the main research questions. Looking back at it, it was not necessary to ask that question, and we could have protected our participants a little bit better by omitting the question in the first place. Threat models are a concept from IT safety, and what it means is that we can think about different ways by which the participant could be re-identified. For example, can the participant identify themselves? The participant typically knows a lot of things, like the date at which they came to undergo the neuroimaging. On the other hand, participants typically have the right to know the data that we have about them anyway. So in that case, what's the problem? Well, you can imagine some edge cases. For example, if the data set contains health information that has not been communicated to the participant. In such a case, there may be a risk if the participant is able to identify themselves. Another threat model is a targeted re-identification, where someone is looking for a particular participant for some reason. A third threat model is a mass re-identification, where datasets are compared on a large scale and an algorithm is looking for matchings. We need to consider, in assessing risk, those technological possibilities that exist today, as well as those that may exist in the future. And therefore it helps a lot to think about the problem from the point of view of information. What information is there in the dataset that could possibly be matched? And so for a matching to occur, we need to have our target population, that's the research dataset that we've got, that may include brain images, and a reference population with some other data to which the dataset could be matched. So if a participant is unique in the target population and in the reference population, then it's possible to make a matching. Now, because of the nature of research data, participants are nearly always unique in the target population. We typically have high-dimensional measures. We have higher precision in the data compared to the number of participants. But it's usually very difficult to find a reference population. And that is a key point. So in considering different kinds of neuroimaging data, for example, if we have anatomical images of the brain, I have illustrated here with an image of my own brain, it's possible to make a matching to other structural brain images. But where could we find other structural brain images? Possibly in a data set of medical records, but those are typically protected by technical and administrative safeguards. Possibly in another research dataset, and so on. There have been several studies of the re-identification risk of structural and functional MRI data, but notably they always assume that the participant is known to be present in both datasets that are being compared. This strongly inflates the risk. Briefly on synthetic data. This is a promising way to share data that do not contain any personal information, provided that they have been synthesized in a way that does not carry personal information further. For example, I could construct a very bad data generating procedure that always outputs my own brain. In that case, the simulation procedure would not be sufficiently safe. However, the usefulness of synthetic data is probably primarily for exploration and model building, since in an inferential step we need to know that conclusions drawn are based on features that occur in the data and not that have been introduced by the simulation. I have some recommendations for information to participants, primarily to respect their autonomy and to some extent to reduce the risk of harm. Here are some suggested phrases to use and to avoid, and I cannot highly enough recommend the Open Brain Consent template, which has been translated to many languages. Finally, here are some points that we may be able to touch on in the coming discussion. How can we make it rewarding for scientists to share their data? How can we move the assignment of credit from scientific papers to a greater variety of research products, including data? What can we do about regulatory obstacles such as the fear of the GDPR to enable data sharing across borders and to find a consensus on what constitutes personal data under EU regulations? And lastly, will it become possible to use federated analyses where data stay in place and results of statistical analyses are outputted freely and shared across borders? Who could operate and maintain such an infrastructure? That's all for me. Thank you very much.
Hello, folks. I believe we are now live. Thank you all so much. There was a lot of ground and perhaps various hills and mountains that was covered in those talks. We heard about annotation, curation, standards development, reuse, and ethics. So I've got plenty of fodder to go on for the next 20 minutes, and we'll take some questions in from the audience as well. Let me start off just by asking each of you to kind of describe what you might want to tell your younger self in terms of fairness. Now, fair, as we know today, is very different given different stages of careers that each of you have. And so what would you tell when, let's say, each one of you started as a student or started doing research, looking back over the years? So I'm going to go in the order of the talks. I'm going to start with Dave.
I thought you would start with you. Yeah, so when I started, so looking back, I think I would begin, before I did anything with data, I would think about, how am I going to organize this data? How am I going to make sure I understand my data in 5 years or even 2 years later in my PhD program? And when I sit down to write my scripts, I would, really be commenting things. Every line of code I write now, or almost every line, I have a comment so that even when I go back, I can remember what was I even thinking here when I, put together this line of code. And yeah, that would be those would be the couple things that I would do. And even now, when I start a research project, I often start collecting data, and then I download the data from a database or wherever it is, the imaging center, and I, put it on disk, and I start going along and thinking, okay, well, how am I going to, work with this data or analyze it? And then I have to stop myself and say, okay, start at step one, organize it and do it, the right directory structure, whether that be bids or whatnot, and start thinking about what I'm going to do for my analyses. Yeah, and from guidance from Satra and Reprint and others, even go one step farther, think about where that code is going to run computationally, make myself a Docker image, run it on that, because later, a year from now, when I write my publication, I'm going to have to make all this available, and building the Docker container at the end after I already did the analyses, not the best step. So those are the things I learned lately.
Thanks, Dave. So moving on to, let's start with Julie first. We'll go in the same exact order.
First of all, thanks for having us. We really appreciate you guys inviting us to this. And I definitely going to say this because I feel much more of a newbie in this field than an expert. So everything I say, I guess I absorbed everyone's talks. Definitely in the context of the Neuromod project, I think the thing that I would tell myself is I thought the hard part was going to be putting together the team and collecting the data and getting stuff in the scanner and the logistics. And although that is challenging, really sharing the data and setting, because everyone else, when they share their data, it makes it look so easy. And the groundwork, at least at our institute, that we're having to do to be able to try and share our data is really surprising. So I'd say start early. That should be on the top priority when you're starting your data set. It's not an afterthought for sure. And so hit the ground running and start, from the beginning, talking to your institution about sharing your data.
Vale?
I guess I'm gonna echo a bit Julie, but definitely what I would tell my younger self is that you should not feel an outsider, and you should not feel like there's someone out there that has the secret recipe and the right talent to make it all work and to make it perfect. Because I did have that feeling, and it did stop me and prevent me from attempting things, just thinking that I could not get them done right. And then there's that mantra when you really learn that you cannot let the perfect be the enemy of the good, and you just start doing it and doing it good enough, good as you can, and it's possible and it's okay. That was a huge step for me.
Wonderful. And I'll come back to mountains and hills later on. But next up, Melanie.
So I actually have a background in theoretical particle physics. So I think the first thing
that I would tell myself when I started my PhD in medical imaging was go even better on the coding part, go know the news tools, because that was my steepest hill to climb, to switch from working on a piece of paper to code and do it properly. So I think all my master students now, I'm saying, hey, get a GitHub account. We're putting this online. Like, have them start early, and also in the education. I think that's what I would tell my younger self.
Thanks, Mel. Elizabeth.
Yeah, no, I mean, I think it's one of those things where you're always learning. So I'm sure in like 2 years, I'll have a completely different answer and I'll be like, oh my God, how did I not know this? But I guess what I would say for now is echoing, I think, what a lot of people have said, this idea of being open by design. So when you start your experiments or when you start your analyses, planning as if you could make it open later. I think that was something where when I was first starting, there were some, like in my master's, there were some experiments I was working on where I was like, I know this data won't be able to go openly available. So I just sort of didn't do it. And it was such a mistake. I could have used so many more FAIR principles, and I could have brought in so much more open source code, and I could have really made my life easier by really adopting this open by design idea. So now I think it's really, really useful just to say, you know, when it comes to the question of can we share or not, like that's hugely important and should be thought about throughout. But adopting, I think what other people have said, adopting as much as you can, as early as you can, really serves not only to benefit the field, but at least in my experience, really serves to benefit you as a researcher and the science you're doing. So I wish I'd known that at the start, but I'm glad I got there a little bit late.
Oh, wonderful. And Gustav.
Yeah, thanks for having us on this panel. So I've been trying to work with data sharing and sharing other research objects for a bit over 10 years. And there's been a remarkable growth of resources during that time. And my advice is to try and find the community. Find your colleagues that are working on new solutions and figure out how you can learn them. So for me, that has been in social media and in places like the Hack Room of the Human Brain Mapping conferences. That's where I found the people that I was able to learn from, how to use new tools and make my data more fair.
Yeah, I think community plays a huge role. So I'm going to take a question from the community over here. So Tom Gillespie asks, do we have an obligation as part of informed consent to educate participants about threat models and the relative risk of those in comparison to the diseases and disorders that we are trying to study? Do we even have to engage in this kind of comparison, or do we inform and allow participants to make their own informed decisions? So I believe this kind of stems from Gustav's talk a little bit. So I'm going to let you kind of respond to this and then let anybody else, especially Julia and Vale, at least from the CNeuromod perspective, kind of talk about it. So go ahead, Gustav.
Right. So thanks for this question. We need to inform our participants as comprehensively as we can in order to give them the highest degree of autonomy in deciding whether or not to participate. At the same time, I think educating participants might be going too far. Maybe it's partly semantic, but if you give people very long forms, they will probably not read them. Of course, if you have an experimental setting where you can sit down and really talk things through with them, then that helps. But the main point, in my view, should be that we make a convincing, we explain convincingly that we as researchers have made an assessment of the risk and come to the conclusion that that it's justifiable. I think that's how I'd answer that question.
Anyone else? Yeah, go for it.
Maybe I would just add, and this actually is more pertinent to my previous experience in a clinical setting, that I agree, like it's not your place to educate, like you couldn't, even if you wanted. but I feel a responsibility to be available when further informations are requested by the participants. So I do believe that there is at least one person has to be available for to be contacted even months after the experiment if further informations are requested by the participants, because that can come out later, especially in clinical setting where news might change quickly and long term.
So for the CNeuromod project, it's a particular project because we only have 6 participants. So actually, Gustav, listening to your talk, I was empathetic to the idea of like subjects either identifying themselves, because if a participant comes out and identifies themselves, then it becomes a 1 out of 5 chance of guessing the others, you know? So in our case, because we have so much contact with the participants, we actually had a lot of discussions with participants around risk, in particular in terms of acquiring this much data on one individual's brain and what the potential of that could be, because we don't actually know in terms of using it to train artificial neural nets. It could be nothing, it could be everything. And so we've had a lot of conversations with participants about risk and tried to deal, I guess, with their questions. And that's definitely one of the reasons we've gone with a registered access. We're not really sure what this data set could have the potential to be. Either it could be, you know, just another neuroimaging data set, like it could be something else. So, and we will review that. And so actually, as people apply to use the data, like if it doesn't fall within the original consent, participants will have the options to reconsent. Say if someone wants to use it to do an art project, they will have the option to be reconsented for that particular project. And so we have a kind of a, we're lucky that our participants are recurrent, there for a long period of time, and that we have that kind of relationship with them.
Thank you. And Dave, you have your hand up. Yeah.
Yeah, I was just going to make a comment. So I do PET research here in the United States. And so we're injecting radiation. And so there's a risk of lots of things. And I think that the IRBs, at least in the United States, that I've seen, do a pretty good job of requiring the investigators to put, you know, all the potential risks in the informed consent. And when we consent our participants, you know, we have someone go through the consent with them step by step, even if they don't read it and discuss those threats. But in some cases, you know, like PET research, you know, the extent of the threat is unknown. The probability that you might develop a cancer in the future because you participated in the PET study has to be balanced with how long did you spend in the sun, what's your genetics. I mean, there's a lot to it. And so, yeah, I think it's our responsibility to go through with the participant, the informed consent and the risks as they are. I think the IRBs are doing a pretty good job of requiring us investigators to list all the potential threats, at least in the United States. So I wouldn't really I wouldn't go farther than they are now, I don't think.
Mel?
Yeah, I would totally echo what Dave has said regarding the PET. I mean, I work on one of the clinical PET sites in Europe, and that is always a discussion point, and we go through it quite detailed as well. That's one of the requests on the European side as well. I would say especially for Gustav and me sitting on this side of the pond, GDPR is inherently protecting our participants from a lot of outside threats, because it just limits what we can do without registered data sharing and so on. And I don't think that, for example, the relative risk of a malicious attack on the server, for example, and accessing all of the data, and then a previously released data set matching and so on, I don't think I would actually take that up with a participant, as Tom was relying to, or I would say thinking about. But of course, I mean, we should be available, like Vale said, for questions at any time.
Oh, one more comment. In our informed consent, probably yours too, it does say that your data is not safe if it's being deposited. I mean, it's not guaranteed to be safe. It's deposited in a database for sharing, you know, that sort of language. And so we do alert them that the data could be lost, but it'll be identified, that sort of thing.
So we have one more question from the audience, and this specifically focuses on the BIDS world a little bit. Has anyone checked how accepted BIDS is in neuroimaging, and potentially can someone comment how good BIDS is now. I might ask Anita for clarification on what that last part means. I could start off very briefly by saying, given some of the uses of some of the tools built on BIDS, we know that it's being used around the world. I don't know if we have an exact number on who all are using it, but maybe that's a good place to start. And I'm going to punt it to both Elizabeth and Melanie for their communities in some sense of using and reusing or building tools on top of BIDS as well. Elizabeth, go for it.
I was going to say, yeah, no. So I think I would agree exactly what you said, Satra, that I think based on the tool usage, so I'm most familiar with fMRI Prep in particular, which is a tool originally developed in the Poldrack lab. And that has seen really broad uptake and adoption based on some of the Sentry reports back on its usage. And so it seems like that has been a success story and speaks to BIDS as a success story. I can say that in my own work, in reusing datasets, really have been focusing on reusing BIDS datasets because they really facilitate reuse in many ways. So for the most part, I would say BIDS has been pretty broadly accepted in that sense. As to how good it is now, I think there are still pieces that are missing. So one I alluded to is like some of the naturalistic stimuli. That's something that's, I think, really interesting and a lot of groups are moving towards, and we don't have great ways to describe that in BIDS if it's not already accompanying the data. And I think there are other kinds of metadata like that I know NIDM is really well placed to help us think about in the future. But Melanie, I know that you've been working on, well, BIDS, the steering committee, and extending BIDS as well.
Yes, and I would say so. I should be better prepared as a member of the steering group, but we have lists with, like, how many citations the BIDS have, how many active data sets are there in BIDS and so on. We have stats about that, and it is really branching out, especially beyond the MRI and fMRI community. EEG is a very active field of research, IEEG also. Now we're trying to add PET, and here Dave can comment on this in a bit as well. PET data is very expensive, and the funding agencies are saying nowadays, you don't get away with having a $5,000 scan lie on your lab's disk anymore. That's simply a waste of taxpayer money. So I think the incentive for sharing and us building the BIDS PET now, and also building OpenNeuroPET as a place to share the data, I think that's really important, because we are just not going to get around it. And there are a lot of different, I think, opportunities to still improve BIDS. I completely agree. Like there are still little things. I'm still hoping for Satra, for example, to really get the provenance extension up and finished. I think that's one of the things I'm looking forward to. And yes, I mean, I think you should just give it a try. Go back to the starter kit. The starter kit has developed quite a bit. So if you've had a look at it a couple of years ago, go back to the starter kit now and give it a try.
Dave.
Good. I know we're running out of time. I just wanted to say, all of us, I'm an old guy, right? So all of us prior to BIDS, long time ago, we all have structured data in our labs. We structure it in some way. We create some folders and files. I think, when we first started talking about BIDS, it was like, why wasn't this done 20 years ago? Because, it is early in its development relative to the span of time most of us have been doing research. It's improving it all the time, and things are getting easier to get data into BIDS. So I think that, and many labs are still using their scripts from their old directory structures. And so the uptake is a little bit slow for labs that have been doing things a really long time. For new PhD students who are collecting new data sets, I think a lot of them are going straight into BIDS for use of the BIDS apps, carrot and stick kind of thing. So I would say stick with it. This is what we need, at least in the field of neuroimaging, to share data sets. I mean, I think it's important to start structuring your data, and the easiest way to get started in neuroimaging is BIDS.
So we have a few more questions, and I know we won't be able to cover. So for the audience listening out here, please feel free to reach out to the speakers and ask the questions. I'm going to kind of merge a couple of questions and kind of extend this BIDS theme and community a little bit. As you know, for many things we do in neuroscience, you become part of a community and the knowledge kind of increases and internalizes as part of it. For someone coming from outside, the BID starter kit was mentioned, but that can still be a daunting task given how extensive the BID specification is today. How would you tell that new person this is how you can participate in this ecosystem, which sometimes for people outside seems like a huge mountain to climb to get to the other side? Whoever wants to go first.
I can start maybe. Yeah, so I think that this is a hugely important question, and it's one I'm very sympathetic to. I really love the idea. I think Dr. Christina Bergman proposed the metaphor of like the open science buffet, which is the idea that, you know, you can think about all of these different practices supporting open data or fair data in particular as kind of a buffet, and you can pick and choose what you need over time. And I think it goes back to the point we made earlier where you don't need to do everything at once. But in particular, with getting started, I think that's a really valuable mindset, because I know in many places, your community, your immediate community is going to dictate a lot of your experience. So for example, if you're in a lab that codes in R, it probably doesn't make sense to go and learn Julia and only code by yourself in Julia when everyone around you is in R, right? And so in a similar way, having that initial base of your own immediate community, I think is really powerful. And then reaching out to the broader community as you hit pieces that you need or you realize are interesting. So for example, one thing I've seen a lot is like folks are really interested in starting to code more in Python, let's say, but they don't have great support in their lab for that language. And so starting to contribute to the neuroimaging and Python ecosystem by participating in open source development can be a really nice way to both get involved and make connections with that community to get that support, and also get feedback on improving your own coding skills in Python. Small interactions are really useful to try and
Thank you.
happen.
And I will put a plugin together with that for NeuroStars and INCF platform to ask questions. And no question is small or big. You should be able to come out and ask any question that you have and help the community kind of help you with that. Anyone else? We are running a little short on time. Yes, Melanie.
Yeah, I was actually introduced to BIDS when I attended the NeuroHack Week. So especially as like PhD students or postdocs, I think it's very cool to go to one of the events. I mean, if you're attending OHBM, go to the Open Science Room. You know, it used to be in person. Now it's not in person anymore, but you can still attend it. And I think there you just meet a lot of people. You get exposed to this buffet of open science, as Elizabeth talked about it. And that's just, I think that's a way of going forward and looking to see what's out there and then getting involved in something that fits your needs.
Thank you. So I'm going to now just bridge a little higher from BIDS to FAIR, now kind of putting the circle back. And we have a couple of minutes to tie the ribbon around this session. So FAIR, like BIDS, or BIDS as we discussed earlier, makes many things fair, but it can still be resource intensive for many people. So some people are looking for tools to introspect their data, and the ecosystem in neuroimaging has grown by leaps and bounds. And so today, I think even F in findability of FAIR becomes an issue as to what you're looking for. So could we just briefly go around with your thoughts on the different components? And you can pick a letter or a couple of letters of FAIR and say how you would advise people listening to you to approach FAIR in the research that they do. So I'm going to randomly pick names now. And I'm going to start with Vale.
Wow, that's a very intense question. I don't know which one I would take. As you said, it's all a process and it's changing. It's a live organism that is changing and growing. And I myself feel like I'm still learning about each of those letters every single day. Right now, because of Neuroscience, I've been mostly like sort of narrowly minded focus on the accessible part, so how to make it open and free. Really thinking of all the pieces of the puzzle, even of just this one word, from the stimuli that, as Julie and Elizabeth pointed out, is not as easy as it seems, to the data itself, which we all understand is harder. Yeah, focusing on that, there's definitely more that needs to be done in even agreeing on what we mean by open and by free, how we give access and to what. But yeah, sorry, I probably missed the question, but those were my last 2 cents.
That's good. Access, and we need to think about it. All right. Talking of that, next person, Elizabeth.
Oh gosh. Okay. I think one thing for findability is I think there's been a lot of really nice work with developing these centralized platforms. So for example, OpenNeuro is huge. I think OpenNeuroPET, I think, will be hugely valuable as well. I also recently have started to use more aggressively this tool DataLab to actually search for data sets that have different characteristics that I need. And I would recommend that as well. I think that's another really nice power of moving to fair data, is that the findability kind of becomes a force multiplier and you can discover more and more data that's really useful for your work. So I would recommend checking out tools like DataLab as well.
Thank you. Keator.
Think about reusability. Think about how to, you can use our tools or others. Think about documenting your data and your methods for reusability. Don't think about your future career aspirations or getting a lot of grant money. Think about your contribution to global science. And in my mind, that's reusability of the data. And it's one aspect, reusability of the data. So you can later on, you can figure out a place to share your data. If you've made it reusable from the get-go, data and scripts, then you're, say, 75% there.
Thank you. Gustav.
And the grant money will come. It's hard to speculate about the future, but I think we're seeing a greater appreciation of fair data outputs. I've just in the last days reviewed a proposal of criteria for fair research data, and it was a long set of criteria, very detailed. I suggested that it's that the criteria are fine, but that they don't serve very well for communication directly to researchers. Give easier advice to researchers. For example, use a good data standard and use a good repository, and that takes you most of the way there.
Awesome. Julie?
Definitely in terms of accessibility, that's something we think a lot about at Neuromod. But in terms of reusability, one of the things that we've done at Neuromod, because we have a really great neuroinformatics team, of which Elizabeth and Valentina contribute to on a regular basis, but trying to take things and the tools that we implement at Neuromod, and we actually have started implementing those in our local scanner to make them available to our neuroimaging community. So maybe people who wouldn't necessarily have the funds to hire a software developer, we take everything and then we put it into pipelines and make it much more easy for them to implement these tools. So it's like, it makes the barrier to entry much less expensive and much less time consuming, so that hopefully more people join this wave.
Wonderful, thank you. And Mel?
So I totally echo what Dave said regarding the reusability. I was lucky to come into a lab where there was a lot of focus on this already. And now, especially with regards to OpenNeuroPET, I think we really focus on the findability and accessibility, because as we said, PET is such a valuable resource, and it's not shared to the same extent as MR or fMRI yet. So I think making it findable, and we'll actually use DataLab in order to make it searchable through the different sites, the American and the European one. And I think that will make a step forward and will maybe also open the other communities' eyes onto, oh, look, there's a data set with PET, fMRI, and EEG. Now we can go and do fun things.
So I know we're a little over time. So I'm going to thank all of you for a wonderful talk and a panel discussion. I know we could probably keep talking for the rest of the afternoon, if not the rest of the week, on these topics. I would encourage all of you and all of you watching to continue this discussion on the forums on the Brella platform, as well as on Neurostars and elsewhere on communities. I think a lot of these questions, and we will try to answer the questions that we could not directly answer on some of those platforms. But these conversations, I think, need to kind of continue and be a bridge for all the people who haven't yet quite gotten to FAIR, but are trying to figure out how to do so. So with that, thank you again, and thanks for participating.